For many years, research scientists have been searching for ways to educate and to vitalize the natural defensive system in our bodies called the immune system. Never has this quest been more important than now. It is accepted that we are losing the war against infectious disease. Many medical scientists and practitioners have resigned themselves to the fact that it is not if, but when the next worldwide pandemic strikes, for which we will have no protection. Because our ability to produce more powerful antibiotics and antivirals is being overtaken by the speed which the germs that threaten us have demonstrated in producing new and ever more lethal strains. Around the world, literally billions have been spent on the research and treatment of cancer, yet the death rate hasn't significantly changed. Autoimmune diseases and allergies are becoming accepted as almost the norm because of the increasing number of people who suffer from these conditions. So what must we do? Stand idly by and accept as inevitable that we are increasingly vulnerable? Fortunately, because of new discoveries that you need to know about, the answer is no. We don't have to accept such an outcome. We can empower our immune systems in order to give us and our family and friends and everyone we can share this information with the optimal opportunity to live the long and healthy life that we desire and deserve. Because of transfer factors. Transfer factors are nature's gift to us. They've been available to us for as long as life has existed on this earth. But only recently have we discovered their empowering role for our immune systems. In 1949, a researcher in South Carolina named H. Sherwood Lawrence was trying to understand how a person can become immune to a disease, how one's body can recognize and respond to a threat without becoming ill from that threat. So Dr. Lawrence found a lab assistant who had been exposed to tuberculosis. He knew this because that individual, when given something called a tuberculin skin test, reacted positively by whelping up in the area of the TB skin test injection. And then Dr. Lawrence tested other assistants until he found one who had a negative TB skin test, which meant that individual had never been exposed to tuberculosis. So Dr. Lawrence drew some blood from the TB skin test positive individual and processed it by spinning it in a centrifuge to separate the white blood cells, the germ-fighting cells called lymphocytes, from the red blood cells. And he took the white blood cells and by further processing broke their cell walls and removed the fluid and collected the fluid from within those fractured lymphocytes. And he put the fluid into a syringe and he injected it into the assistant who had never been exposed to tuberculosis and waited. It was really a pretty nervy thing to do. 2 days later, he did a TB skin test on the assistant who had never been exposed to tuberculosis. And lo and behold, the skin test whelped up and he or she reacted just as if they had been exposed to tuberculosis. Dr. Lawrence had transferred the recognition of the tuberculosis germ from one who had been exposed to tuberculosis to one who had never been exposed. He didn't understand exactly what had happened. He just knew he had transferred something, some factor from one individual to another, and by doing so, he had transferred the ability to recognize a germ, tuberculosis, in an individual who hadn't had prior exposure. And thus the term transfer factor was introduced into the scientific world. Over the next several decades, millions of dollars were spent on transfer factor research, and over 3,000 papers were written. But the process of producing transfer factors was very expensive and time-consuming. Use of transfer factors was mainly confined to the lab, and the public was unaware of the potential health benefits that transfer factors offered. In 1989, 2 researchers in South Carolina made a very important discovery. Doctors Wilson and Paddock found that transfer factors were abundant in the first maternal milk called colostrum, that's produced right at and right after birth. Further research revealed that all mammals produce transfer factors that are molecularly identical. It was discovered that Mother Nature has devised a method for the mother to pass on her immune system knowledge to her newborn by nursing. This first benevolent act by the mother enables the newborn, thrust from a warm, sterile environment of the womb to a harsh, cold world filled with germs, to survive. Let's examine exactly how this happens. Chickenpox is a good example to use because before the chickenpox vaccine, most people contracted this viral illness. But extremely rarely did anyone have a second bout with chickenpox. Why? Why not develop the illness over and over? Because the individual may have been exposed to chickenpox dozens, hundreds of times after that first episode. On the screen, you'll see a diagram which demonstrates how one builds up immunity to a disease. The straight line highlighted is a time sequence from the exposure to a disease until the immune system begins to respond. In this instance, it would be while the person with chickenpox was breaking out in little red bumps, which transformed to multiple itchy blisters. After several days, the immune system cells, which you see highlighted, begin to multiply. And once sufficient immune system cells have been created, you begin to win the battle, and the chickenpox virus is defeated. The blisters scab over, and eventually you get well. Your immune system ultimately protected you, but not before you went through some pretty miserable days of illness. So what then occurred? Your immune system stored the memory of the chickenpox virus in special immune system cells, which we refer to as, quite appropriately, memory cells. Later, you can walk into a room full of people with chickenpox and remain totally unconcerned because now your body's immune system has knowledge about the identifying features of the chickenpox virus. And so, even though the chickenpox virus frequently tried to attack you again, it was unsuccessful because each time it entered your body, the particular immune system memory cell that carried the identity of the virus instantly alerted its fellow immune system cells, and a huge number of fighters was produced, which overwhelmed the hapless chickenpox virus. And you didn't even know that you'd been exposed. You never became ill. You can see the short line in what is called a secondary immune response as compared to the time sequence when you became ill, that primary immune response. This makes sense, doesn't it? What would be our quality of life if we became ill over and over with the same disease? We'd be ill all of the time. But by having an immune system that can remember prior illnesses, we can develop what is called resistance or immunity to that disease. And what we've discovered is that the little protective memory molecule that provides us protection is that factor that Dr. Lawrence transferred from the person who had been exposed to tuberculosis to the person who hadn't been exposed. Memory molecules, which give us early recognition and a quick response to a disease threat, are called transfer factors. Wouldn't it be wonderful if we had a factory containing machines where we could produce transfer factors so that we could consume them and have them in our bodies without having to go through the misery of having a disease like chickenpox? Well, the good news is that such factories exist. They are called dairy farms or hen houses. And the machines? Milk-producing cows and egg-laying chickens. Dr. Richard Bennett is a researcher who's been very involved in the issues that affect the dairy farm industry. Dr. Bennett has a Ph.D. in comparative pathology from the University of California, Davis, and has been a consultant to the prestigious National Research Council, and has served as an advisor to the FDA, the USDA, and many state and local agencies. Dr. Bennett is recognized and respected as an authority on something called colostrum. Colostrum is the first milk that a mammal produces right at and after giving birth. It is different than later milk in that it is thicker and more yellow. And even though it was known that colostrum provides nourishment, Dr. Bennett began to formulate theories on exactly what else this colostral production provided the newborn. His research led him to analyze mammary or breast tissue during the birthing and colostral period. And he noticed, when analyzing the breast tissue under a microscope, that there was a cascading of immune system white blood cells that were flooding into the breast tissue. These immune system cells were germ-fighting cells called lymphocytes. What were they doing there? Germ-fighting cells are supposed to go after germs. But there was no infection in the breast tissue. Over the years, Dr. Bennett discovered, as did other researchers, that the lymphocytes were there for a life-sustaining reason. They were depositing the mother's immune system knowledge into the breast tissue so that knowledge could become part of the colostrum. And when the newborn nursed, it would receive the important immune system knowledge that the mother had accumulated over her lifetime. The mother was, here's that term again, transferring something into the newborn. And now we know that she was transferring transfer factors. Transfer factors are memory molecules of immune system battles fought and won, and memories stored. They are produced by those germ-fighting cells called lymphocytes. In the animal kingdom, if a newborn doesn't immediately nurse, it often doesn't survive. And if it does survive, it often doesn't thrive. Humans operate exactly the same way. But fortunately, even if our human newborn doesn't nurse, it is often kept out of harm's way by being given sterilized water and formula, and by being bathed and kept in a clean crib until its immune system, totally naive at birth, has been given time to begin to develop. So what does this all mean to us? Again, over the last 50 years, researchers have slowly but steadily unraveled the importance and characteristics of transfer factors. They have discovered the following. A. All animals produce transfer factors, not only mammals, but other species like birds. B. All transfer factors are molecularly identical, no matter what species produces them. This is very important to remember. If a cow produces a transfer factor for a germ like E. coli, and a human produces a transfer factor for E. coli, they are going to be of the same molecular structure. The same holds true for a chicken. Same germ E. coli, same transfer factor structure. C. Transfer factors are very small, and through trial and error, it has been discovered what their size is, so that by developing the right size pores in a filter, they can be separated from colostrum. Or, if the source of transfer factors is from a chicken, they can be separated from the yolk of an egg. D. Transfer factors can be given by mouth. Dr. Charles Kirkpatrick, a physician and researcher in Denver, Colorado, has published a paper in the journal Biotherapy that documents that the oral route is the preferred method of administering transfer factors. We now know that the regular consumption of transfer factors can provide protection through early recognition of threatening germs, just like having had chickenpox gives one the ability to recognize chickenpox and to have resistance. Transfer factors can provide us that same type of benefit. Transfer factors have been shown to have additional benefits. There is an immune system cell that is on the front line of our defensive system. It is called a natural killer cell. Don't you love that name? Natural killer cells are effective in seeking out viruses, in seeking out intracellular bacteria, and in identifying and attacking abnormal cells that can turn into cancers. A report from Rockefeller University issued in February of 2004 stated that natural killer cells can be the body's new gun for hire in tumor and infection control therapies. But the researchers said that natural killer cells have to be nurtured. Their ability to destroy tumor and infected cells is not present at birth. The report concluded by saying that this new insight will enable scientists to pursue research into activating natural killer cells to help the body fight emerging infections and tumors. Transfer factor preparations that have been produced from egg yolk and colostrum have been shown to increase the activity of natural killer cells by 283%. This research was recently performed by the Russian Academy of Medical Science. This combination of egg yolk and colostral transfer factors can be made even more effective by adding other ingredients, which have been shown to cause an even greater increase in natural killer cell activity. These ingredients: maitake and shiitake mushrooms, cordyceps, a Chinese fungal extract, inositol hexaphosphate, beta-glucans, mannans from aloe vera, olive leaf extract, a phytosterol from soybean extract, and Agaricus blazei, another mushroom, were added to transfer factors in the Russian Academy tests, and the result was a 437% increase in natural killer cell activity above the baseline. The lead researcher, Dr. Kisielewski, stated that his laboratory used interleukin-2, or IL-2, as the gold standard for comparing combinations of ingredients which could stimulate the immune system, and that this combination of transfer factors and the added ingredients surpassed even interleukin-2. There was a recent report that the Russian Ministry of Health has approved the use of this combination of transfer factors and the added ingredients for doctors to use in hospitals and in their clinics.